Hi, I'm Katherine Lambert.
And I'm Kirsten Littlefield, and today we're going to be presenting on DNA topoisomerase 2 beta.
So when many people think of DNA topoisomerase, they relate it to transcription. While that is true, DNA topoisomerase is much more intricate. There are actually 3 types. There's type 1, 2, and 3. A lot of people don't know that there's 3 types. Only in a high-level college course would you probably find that out. The broad function of DNA topoisomerase is to catalyze the interconversion of topological isomers of DNA and unknot and untangle the DNA strands. This picture gives a good basic overview of where topoisomerase is located on the DNA double helix. You'll see here it does its job from keeping this untangled as the 2 separate strands separate. And then localizing in more on what we're going to be talking about the rest of the presentation, topoisomerase 2, it is located in the nucleus where they're in eukaryotic cells. The function is to alter the tertiary structure of DNA by transient breaks, so breaking the, I don't have a picture up here, but it breaks the double helix. And then it also plays roles in chromosome structure and function. An example is relieving torsional strain during replication. We have a short video here to give a basic understanding of the function in an easy way.
Topoisomerase 2 enzymes do the same thing, but with both strands of the double helix.
So it cleaves the double helix.
Topoisomerase 2 cuts both strands of a double-stranded DNA.
And another double helix will go through.
And then reseals the break.
And then reseals the original double helix that was broken.
DNA is supercoiled.
We predicted that the secondary structure would be a globular protein with a complex function. We predicted multiple subunits consisting of alpha helices, beta sheets, and random coils, and possibly some prosthetic groups throughout.
And moving from primary and secondary structure, we then looked at tertiary structure, which is evident in this image. Here at the protein data bank, we were able to find out that this image is pretty complicated until you look at it through the secondary structure. Here, we were able to see that our predictions were correct. We see alpha helices in pink, yellow for beta pleated sheets, and purple for the DNA around which the enzyme is functioning. Here also, we were able to zoom in and see that when you click on ions, you have 2 magnesium ions per subunit acting with the DNA, supporting our hypothesis with the presence of cofactors. Additionally, we were able to find out that there are 2 isozymes of topoisomerase 2, topoisomerase 2 alpha and topoisomerase 2 beta. From here on out, we'll be focusing on topoisomerase 2 beta. Here, we were able to find out what the function of topoisomerase 2 beta was. It controls the topological states of DNA, which Kat mentioned before, suggesting that there is ATP-dependent breakage of the double-stranded helix, and that was demonstrated in Kat's video previously. Additionally, another double helix is passed through, and then the original DNA helix is rejoined, preceding the function of DNA of topoisomerase 2 beta. Additionally, as you saw in the image previously, there were 2 cofactors per subunit, 2 magnesium 2+ ions per subunit, and they can also be used during the formation of salt bridges within the protein and the DNA. Therefore, also, they can use the divalents magnesium 2+ and calcium 2+. Also, the topoisomerase 2 beta enzyme exhibits a toperin domain, which is approximately 100 amino acids long and has 2 conserved motifs. One conserved glutamate, which we predicted would act in acid-base catalysis, and another conserved motif exhibiting 2 aspartates, which we predict would orient our cofactor magnesium ion. Here, we explored this domain further and found out that its function is catalytic, and it functions in DNA strand breakage and rejoining. Here, we were able to find out that our predicted functions of the 2 conserved motifs were correct. Our conserved glutamate acted as a general base in nucleotide polymerization and strand rejoining, but as a general acid during strand cleavage. Also, our 2 conserved aspartates were able to coordinate our magnesium cofactors that are required for topoisomerase activity.
So we know that topoisomerase II cleaves DNA. As we saw in the video, it cuts both strands of the double helix very cleanly. But how exactly does it know where to cleave the DNA? Well, unfortunately, sequence recognition accuracy is really low. But luckily, there's a general pattern of the cleavage sites in a DNA molecule. They recently found a TATA sequence, and that really helped improve the enzyme specificity. And when DNA cleavage is happening, the polarity is at the 5' end. Here we have a nice, complete picture of the mechanism of DNA topoisomerase II. The different colors represent the different domains. The two I'll be touching on the most will be the yellow ATPase domain and the blue DNA cleavage domain. So you'll see here the DNA double helix will come in the cleavage domain, and the 2 ATP will bind, ATP hydrolysis will take place, and the 2 ATP will allow the other double helix to come through, and as that happens, the first double helix that was brought in will cleave, goes through the second double helix, the complete one will continue going through the topoisomerase, and then once that is gone through, the first double helix that we talked about that was cleaved will religate, the products will be released, and the process will begin again.
And as we know, the function of an enzyme is dependent on its amino acid sequence. And here we were able to find out that our amino acid sequence for topoisomerase II beta is approximately 1,620 amino acids long, and within that sequence there are several sites. 2 being ATP binding sites, which are essential to cleave ATP and also support the function that we just saw previously. Also, 3 metal binding sites were found within this amino acid sequence, which are essential to binding our cofactors, magnesium ions, and also an active site, which is essential for the breakage of DNA, and a DNA binding site, which facilitates the breaking of the DNA double helix. In our research, using UniPro and Ecstasy, we were able to do a blast on our topoisomerase II beta, which allows us to identify sequences that are very similar to that which we are studying. We found that a 99.6 identity match existed between topoisomerase II beta in humans and topoisomerase II in pan troglodytes, which is another name for the common chimpanzee. Additionally, there was a 99.1 identity match for topoisomerase II in rhesus monkeys. However, knowing that we are very similar to monkeys, we were wondering how similar is our topoisomerase II beta to our topoisomerase II alpha. And here we can see that there's a 68.2% identity match, suggesting that the 2 isozymes are less common than human topoisomerase II to topoisomerase II in monkeys.
So moving on to our primary source, we chose a paper that did a good job representing what we wanted to talk about because it showed the differences between topoisomerase II alpha and beta, and also the differences between topoisomerase II beta 1 and topoisomerase II beta 2. And because the human cells have 2 distinct types of topoisomerase II isozymes, alpha and beta, and other proteins that are homologous to the topoisomerase II in human cells are Drosophila, yeast, and bacteria. And most amino acid sequence divergence from the human cells to the Drosophila, yeast, and bacteria occur at the C-terminal and N-terminal domains at the end.
Also, what we found during this study was that there are 2 distinct splice variants of topoisomerase II beta, which Kat mentioned previously, beta 1 and beta 2. These are resulting from differential splicing within the topoisomerase II beta gene.
Moving on to the methods and materials of our primary source. So first, to isolate topoisomerase II beta cDNAs, they used agarose gel, and then only the DNA in size ranges 9 to 15 were excised from the gel to separate the topoisomerase II beta cDNAs and alpha cDNAs. They were purified using Gene Clean 2. and the cDNA clones were sequenced using sequenase, and the topoisomerase II alpha and beta probes were paired by PCR, which is a polymerase chain reaction, and a polymerase chain reaction amplifies a specific segment of DNA. SDS was also used in the methods and materials part of this primary source and I wanted to mention that because we talked about that a lot over the semester in class.
All right, and then moving into the results from our paper, they identified cDNA clones encoding this variant strand form of topoisomerase II beta, which we saw previously with the beta 1 and beta 2. How they did this, they went and screened human B cell cDNA libraries using a topoisomerase II beta sequence as a probe, and found that one clone had an insertion near its 5' end, and that was what they called topoisomerase II beta 2. Then they analyzed the genomic DNA of topoisomerase II beta gene in order to distinguish between these two variants. They compared the sequences of beta 1 and beta 2 cDNAs and found where the insertion was inserted or extracted from the region in the topoisomerase II beta gene. Here they found that two splice acceptor sites separated by 15 base pairs. With further testing, they found out that 15 base pair section was actually how big the insertion in the 2 beta 2 gene was, and this encodes 5 amino acids. These 5 amino acids could have two potential functions: one to disrupt an important domain, which would distinguish beta 1 and beta 2 variants, or also it could introduce key residues conferring additional functions to the topoisomerase II beta 2 variant that are distinguished from the beta 1 variant. In order to assess how these variants are expressed in humans, the scientists in this experiment went about two methods to determine which one was more highly expressed. One was a cDNA library analysis, in which they found one clone per human B cell cDNA library contained the variant. And also they went about an RNase protection analysis, in which topoisomerase II beta probe was hybridized with both beta 1 and beta 2 sequences, and found that given how this probe annealed with the different sequences, there was different fragment sizes due to fragment protection by the probe. So therefore, due to differential annealing, the beta 2 fragment was found to be 296 base pairs long, whereas with the beta 1, it was endonucleically cleaved, which means that its fragment was shorter at 228 base pairs long. Therefore, because these two fragments were different sizes, they could distinguish between which one was being expressed in 7 different cell types. And there they were able to find that there was a 20 times higher expression of beta 1 than beta 2 variants in 7 different cell types.
So to wrap up our presentation, we wanted to relate topoisomerase back to the real world, and we actually came across some information that showed us that topoisomerase inhibitors are really strategic therapy for cancer, and especially cancer chemotherapy. The topoisomerase II inhibitors block the ligation step in the cell cycle and initiate cell death pathways, otherwise known as apoptosis, killing those cells that are cancerous, and therefore being a really great use for cancer therapies for humans nowadays. And we thought this would be a great way to wrap up our presentation. We hope you enjoyed our talk.